So looking through this massive stack of embryology notes and clinical lab protocols you sent over to us, there is this, well, there's a very specific, almost universal misconception that just jumps right out.
Oh, absolutely. It's right there in almost every single paper.
Yeah, and I think it's because when you think about the exact moment a new potential life is, set into motion, it is so easy to picture this incredibly dramatic event.
Right, like a spark.
Exactly, a spark, a high-speed collision, like this microscopic firework going off the exact second a sperm and an egg finally meet.
Which makes sense. I mean, that is exactly the way that moment is always portrayed in documentaries or textbooks. It's shown as the grand arrival.
The triumphant finish line of this very long, arduous race.
Right, but when you actually step into the reality of the protocols you provided and, you immerse yourself in the environment of an actual embryology lab, that entire narrative completely changes.
It totally flips because the lights in the lab are low. The pace is incredibly slow, quiet, and deliberate.
It has to be.
Right. Because according to the biology we are looking at today, that dramatic collision isn't the end of the story at all. Honestly, it is barely the prologue.
Yeah, that's a great way to put it.
So, welcome to the deep dive. For you, our listener, who provided these really fascinating materials, today marks the 7th installment of your Inside the IVF Lab series, which we are calling Did Fertilization Happen? The Pronuclear Check.
And we really want to try and match that careful, deliberate pace of the lab today. Because what we are doing in this deep dive is shifting our focus entirely.
We're zooming in.
Exactly. Up until now, we've been looking at the separate gametes, so the sperm and the egg and the various ways the lab handles them. But today, we are looking for the very first proof point that a viable journey has actually begun.
We're finally asking the big question.
Right. We are exploring the mechanisms of what an embryologist is actually looking for through the microscope and why the critical question changes from, did the gametes meet, to is an embryo actually forming?
Because right out of the gate, your sources make it so clear that we need to bust this massive illusion, which is the idea that insemination automatically equals fertilization.
It does not. Not at all.
Let's unpack the mechanics of this, right? Say you go through a retrieval and the lab performs conventional IVF, so they're mixing the cells in a dish. Or they use ICSI, bypassing the outer shell entirely.
Right, the direct injection.
Yeah. As a patient, it is so tempting to get that update and assume, okay, the sperm is in the egg, we have embryos. But looking at the cellular biology here, you don't. Not yet, anyway.
You really don't, because fertilization is this highly complex sequence of biological events. It is absolutely not a procedural guarantee.
Even with ICSI?
Even with ICSI. I mean, where the embryologist uses a microscopic needle to place a single sperm directly into the oocyte cytoplasm, the egg cytoplasm of the lab is merely facilitating the meeting.
Making the introduction.
Exactly. The cells themselves still have to execute a very precise internal cascade of events.
It really makes me think of planting a garden. If I take a seed and I push it down into the soil and water it, I haven't actually made a plant yet.
Right, you've just done the prep work.
Yeah, I've just created the optimal conditions for one. Just because the seed is sitting in the dirt doesn't mean a green shoot is going to magically sprout. You have to wait.
And then you have to actively look for physical evidence of growth.
Exactly.
And if we take that analogy a step further into the biology, the soil in this case actually has to wake up and participate. The oocyte has to actively recognize the sperm's presence.
Wow. Okay. How does it do that?
Well, when the sperm enters, it brings along a specific protein, and that protein triggers this massive oscillating wave of intracellular calcium that sweeps across the egg.
Like a shockwave.
Basically, yeah. That calcium wave is the biological alarm clock. It tells the egg to resume its metabolic activity, to unpack the tightly coiled DNA inside the sperm head, and, to simultaneously reorganize its own chromosomes.
And none of that internal remodeling is guaranteed just because the sperm breached the cell wall.
No, not at all. Which means the laboratory can never declare fertilization at the moment of insemination. It's an internal biological cascade that must be visually confirmed later.
You have to check. You cannot assume.
Exactly. The process relies entirely on the egg and sperm completing that internal dance on their own.
But the timing of that visual check, it seems incredibly precarious based on the lab schedules in your note.
Oh, it is. It's a very tight window.
Because the embryologist performs this check the morning after insemination, strictly between, 16 and 18 hours after the gametes were introduced. Why such a narrow window? Are they
Yeah.
basically trying to photograph a moving train here?
It is entirely like trying to catch a moving train, and the evidence does in fact vanish if you miss it.
Vanish, like completely disappear.
Yes. So if you pull the dish out of the incubator and look too early, say at 10 hours post-insemination, the biological machinery is still setting up. The specific markers we need to see haven't fully formed yet.
Okay, so too early is no good.
Right. But if you wait until 22 or 24 hours, the evidence you need to confirm normal fertilization has already disappeared.
And the notes explain why it vanishes, which is just fascinating. It's a process called syngamy.
Syngamy, yes. So when fertilization is progressing normally, two distinct structures form inside the egg, and these house the genetic material from the mother and the father.
But they don't just sit there statically, right?
No, they don't. They actually migrate toward each other.
That is so wild. How do they move?
They migrate along microtubule tracks. You can think of these microtubules as like a temporary scaffolding or a cellular railway system inside the egg.
Railway system, okay.
Yeah, and interestingly, the sperm actually brings the organizing center for these tracks. So the maternal and paternal structures are pulled along this railway toward the center of the cell.
And then they meet.
They do. And once they meet, their nuclear envelopes, which are the protective membranes holding them together, they completely dissolve. Yeah. The chromosomes from the egg and the sperm then align together on a shared meiotic spindle, and they prepare for the very first cell division.
So that moment of alignment, that is syngamy.
Exactly. And once those envelopes dissolve, there is literally nothing left to see under the microscope. The structures are just gone.
Which leaves the embryologist looking in a single, uniform cell.
Right. If they missed that strict 16 to 18-hour window, they have absolutely no idea if that cell successfully fertilized and already went through syngamy, or, if the calcium alarm clock never went off and it never fertilized at all.
So the pronuclear check is informative precisely because it captures a highly temporary transition.
It's just a snapshot, yeah.
Okay, so let's talk about the specific evidence captured in that snapshot.
Yeah.
To get a normal fertilization report, which your research highlights as the gold standard for this stage, the embryologist is looking for two distinct things, right? Pronuclei and polar bodies.
Yes. So a pronucleus appears as this tiny, clear, spherical bubble inside the cytoplasm of the egg. And in normal fertilization, we need to see exactly two of them. One maternal pronucleus, housing the egg's chromosomes, and one paternal pronucleus, housing the sperm's chromosomes.
It really makes me think of like a nightclub bouncer checking IDs.
Oh, I like that. How so?
Well, the cell is this exclusive venue we can call the embryo club. And to get in, you need to show exactly two IDs, one from the egg, one from the sperm.
Right.
And if you have those two IDs, you get stamped as a 2PN2 pronuclei, and you're officially categorized as a zygote.
I think that captures the gatekeeping aspect perfectly, and keeping with your analogy, just outside the main body of the egg, but still trapped under its outer shell, the bouncer also needs to see two discarded coats at the coat check.
The discarded coats, those are the polar bodies.
Exactly, because the human egg actually starts its development with twice as much DNA as it needs to combine with a sperm.
Wait, really? Twice as much?
Yeah, it has to shed half of its genetic baggage before it can safely enter the club, so to speak.
So how does it get rid of it?
It goes through what's called an unequal meiotic division. So instead of splitting perfectly in half like a normal cell would, the egg keeps all the cytoplasm and all the nutrients for itself.
To support the future embryo, right?
Exactly. And it physically pinches off this tiny little packet containing just that extra half of its chromosomes.
And that packet is the polar body.
Right. Now, that first polar body is extruded when the egg initially matures, but the second polar body is extruded only after the sperm enters and triggers that calcium wave we discussed earlier.
So the bouncer is looking for two IDs inside and two discarded coats outside.
Precisely. Seeing two pronuclei inside and two polar bodies outside confirms that the entire biological sequence executed flawlessly.
The perfect scenario.
Yeah, the egg kept exactly the right amount of DNA, the sperm successfully unpacked its DNA, and both sets are actively visible. That 2PN status is the beautiful textbook benchmark used to calculate your normal fertilization rate.
But the reality of biology is often messy, isn't it?
Oh, very messy.
Because when the embryologist peers through the microscope at hour 17, they don't always see that perfect textbook picture. And because the timing is so ambiguous, seeing 0 IDs, a 0 PN, that really makes the lab hold its breath.
It does. A 0 PN finding means no pronuclei are visible at the time of observation. Now, it is crucial to understand that this doesn't definitively mean total failure.
Right, because of the moving train.
Exactly. The biological train might just be running slow, meaning the microtubule scaffolding hasn't, pulled the pronuclei into a visible formation yet.
Or it went too fast.
Right. It ran unusually fast, and syngamy already occurred before the embryologist even looked.
But surely seeing something is better than nothing. Like if the embryologist looks in and sees a single pronucleus, a 1PN, does that mean the train is at least partially at the station?
You would think so, but 1PN is actually where things get significantly more dangerous.
Really? Why?
Seeing only one pronucleus often points to a severe biological misfire. It's highly ambiguous.
The notes detail something called parthenogenetic activation here. Is this where the egg basically gets tricked into activating?
That is exactly what happens. Sometimes the oocyte experiences a spontaneous calcium wave without any sperm actually contributing its DNA.
Just out of nowhere.
Yeah, and the egg reacts as if it's been fertilized. It resumes metabolism, forms a single maternal pronucleus, and might even attempt to divide.
There's no dad.
Right, because there is no paternal genetic contribution, it's a developmental dead end. But on the other hand, a 1PN could just be an eclipse.
An eclipse, like they're hiding each other.
Yeah. You mean both pronuclei actually formed, but because the microscope is looking top-down, they are physically stacked on top of each other.
So the paternal ID is hiding right behind the maternal ID.
Along the microscopic axis, yes. And because of this massive difference between a fatal genetic misfire and just a simple visual trick, 1PN zygotes are interpreted with extreme caution.
Wow. Which brings us to the other extreme, right? Seeing too many IDs.
Oh.
A 3PN zygote, 3 pronuclei.
Right, the 3PNs.
In conventional IVF, the mechanism your sources point to is fairly obvious. Polyspermy.
Yes.
2 sperm managed to breach the zona pellucida before the egg could, lock down its outer shell. So you have 2 paternal IDs and 1 maternal ID.
That is the classic cause in standard insemination. Extra genetic material from multiple sperm entering simultaneously. But polyspermy doesn't explain everything we see in the lab.
Wait, hold on. If we look at the ICSI protocols you're referencing, you only inject a single sperm with that microscopic needle.
Correct. Just one.
So the extra DNA literally cannot be paternal. Is the egg failing to drop that second coat at the coat check we talked about?
You hit the nail on the head. This is such a crucial biological concept. Even when ICSI delivers a single sperm perfectly, the egg's internal machinery can malfunction.
Just doesn't work right.
Yeah, the meiotic spindle, that delicate internal structure responsible for separating the chromosomes and pushing that extra half out into a second polar body, that can just fail.
So the egg accidentally retains its extra set of chromosomes.
Yes. And those retained chromosomes then form an additional maternal pronucleus.
That is incredible. So you end up with 1 ID from the single sperm you injected and 2 IDs from the egg just because it failed to throw away its extra baggage.
Exactly.
A 3PN caused entirely by a maternal meiotic error.
It really completely reframes how we view fertilization, doesn't it? It proves that creating a viable zygote depends on much, much more than just the route of sperm entry.
It's not just about getting the sperm inside.
No, it depends heavily on the egg executing a flawless internal dance of chromosomal division. If the egg makes a meiotic error, you get abnormal fertilization, even with a technically perfect ICSI procedure.
That really underscores how much heavy lifting the leukocyte is doing in this process.
Oh, absolutely.
But it also leads us to a very difficult clinical reality. Because these abnormal patterns, the 1PN eclipses, the 3PN meiotic errors, and the true 0PNs where the alarm clock never goes off, they result in the first great narrowing of the IVF cycle.
They do. It is known as the fertilization rate. And for many patients, it is an incredibly distressing hurdle.
Because the numbers just drop.
Right. You start with a certain number of follicles seen on an ultrasound. A smaller number of eggs are actually retrieved during surgery. An even smaller number are mature enough to even attempt insemination.
And then the fertilization check drops the number of viable cells again.
It's arguably one of the most agonizing points because the physical hurdles are done, the stimulations, the retrieval, the gamete handling, that's all finished. And then you just have to sit there and wait and see if the biology cooperates.
Total fertilization failure is just a devastating outcome.
It really is.
Right.
And as the research shows, it can happen after standard IVF or after ICSI.
What causes it usually?
Well, it can stem from severe sperm dysfunction where the sperm lacks the specific proteins needed to trigger that crucial calcium wave. Or it can be oocyte activation failure, where the egg's internal receptors simply don't respond to the sperm's signals.
So the gametes are physically together, but the communication completely fails. But let's look at the best case scenario to balance this out a bit.
Mm.
Let's say you get the call from the lab and your cohort is entirely 2PN.
Best news you can get that day.
Right. You had 10 eggs, and 9 of them have 2 clear pronuclei and 2 polar bodies. You've got the balancer's approval across the board.
Yeah.
The temptation is to celebrate as if the finish line has been crossed.
Oh, of course. And a high 2PN rate is a fantastic, encouraging milestone. The clinical team is thrilled, and you should be thrilled. But based on the data and your sources, we have to strictly manage expectations. A 2PN zygote is simply suitable to continue in culture.
Meaning it has the right starting equipment.
Exactly. It has the proper number of chromosomes, but having the equipment doesn't guarantee the biological machine will actually run.
Because building a human being requires, what, thousands of steps?
Thousands of consecutive steps, yes, functioning flawlessly. The genes must activate at precisely the right moments. The cells have to divide symmetrically and hit complex metabolic milestones.
So a 2PN status cannot guarantee blastocyst development.
No, it doesn't ensure the embryo is chromosomally normal, and it certainly does not guarantee a live birth. It is a strong start. It's not a finished success story.
It's kind of like getting accepted into an incredibly rigorous university. The acceptance letter, the 2PN, is a huge deal, and it proves you have the credentials to be there. But you haven't graduated yet.
You still have to pass all the exams.
Exactly. And this fertilization check is the bridge. It's the moment the process stops being about single, separate gametes and starts being about nurturing embryos in culture.
It is the definitive transition point in the laboratory timeline.
Well, as we close out this installment, I want to sincerely thank you, our listener, for sending over such an incredible stack of clinical resources and for joining us on this slow microscopic journey today.
It's been fascinating.
It really has. The sheer complexity of the shift that happens in just 18 hours is staggering. Yesterday, the lab was focused on handling fragile individual cells. Today, the entire focus has shifted to supporting early embryonic life.
And as we step away from the microscope today, the sources you provided leave us with something I think really fascinating to ponder.
Oh, what's that?
Well, we've established that the pronuclear check is merely a fleeting window, right? A brief 16 to 18-hour snapshot of a rapidly moving biological train.
Right.
The embryologist looks for just a moment to verify those pronuclei and then immediately returns the dish to the safety and darkness of the incubator.
Safe in the dark.
Yeah, so it makes you wonder how much of early human development is happening completely unseen, how much of the very origin of our cellular life takes place entirely in the dark, in the quiet, unobserved hours between the times we choose to look.
Wow. That is a hauntingly beautiful thought. We really are just getting these tiny scheduled glimpses of a cellular machinery at work.
We are.
And speaking of what happens in the dark of those incubators, that is exactly where we're heading next time. In the 8th installment of your series titled Embryo Culture, Creating the Right Environment, we're going to step away from the microscope entirely and go inside the incubator itself.
Getting into the environment.
Yes, we'll explore the culture media, the precise gas control, the pH balancing, and the incredible environmental engineering required to keep those tiny 2PN zygotes alive and developing unseen. Until then, remember, just because the seed is in the soil doesn't mean the work is done. Thanks for taking this deep dive with us.